Good afternoon. Welcome back. We're starting exam 2 material today with lecture 10, the cell cycle. I am Dr. Beal. You will notice I am wearing my chromosome dress, which is more awesome in real life than it is on video. But I'll at least draw attention to the fact that today we're going to be talking about chromosomes and DNA. We're going to talk about how cells replicate themselves. And this is the beginning of the exam 2 set of information. Exam 2 covers mitosis and the cell cycle today, all the way through human genetics. And it's one big idea about DNA and how information gets passed on, particularly in eukaryotes, but a little bit of prokaryotes. Quick reminder, the exam is on Sunday, October the 11th. You got an email from us last week, if you're watching this and you haven't had the exam yet, so a little bit time off. You got an email from us on October the 1st or 2nd, giving you specific times for logging in. Everyone takes the exam at exactly the same time, but we're going to have you log in groups so that we don't crash the Chi. So read the email, come to our reviews this week. We're also going to have a Friday night review or a Friday afternoon review to talk to you a little bit more about exam stuff. Exam 1 covers memory and learning through and including cell respiration. So definitely the cell respiration stuff is on the exam. Memory and learning is on the exam. The exam has 70 questions. You have 85 minutes. It is open notes, but you don't have a lot of time per question. Your best strategy is to review outcomes, these outcomes that we're looking at, not these, the ones for the lectures that are on the exam, but the outcomes for the exam. See if you can do them from memory, identify everything, and then test yourself before the exam. Figure out what you need to focus on, what you don't need to focus on, and then by the time you get to the exam, not only will you be comfortable with what the questions are going to ask, because we don't give you practice questions, we give you the outcomes, we give you what is going to actually be on the exam. You'll be comfortable with what is on the exam, and then you'll be a little bit comfortable with how you take the exam as well. For those of you who have been asking, we don't give you practice questions. The practice questions are what you have been doing in workshop. Those are examples of old exam questions that you've been doing. If you want to know what's on the exam, look at the outcomes. That is literally a list of questions that are going to be on the exam. You should be able to do that with your organizers or from memory in front of you pretty quickly at this point. And I'll see you soon. I'll do this lecture first. I'm ready to get done already. It's Sunday night for me. For cell cycle, we're going to look at chromosomes, chromosome number, chromosome structure. We're going to have to identify all the parts, and then we're going to put them together and do the mitosis dance. This is just the beginning of a longer lecture on how we move chromosomes around. Lecture 11 is meiosis. So I'm going to introduce some ideas in this topic that you're going to take over into the next lecture. So just a little bit of detail here, a little bit of explanation in the next lecture. I'm also going to start providing some suggestions to prepare for what's coming up next. Now we have some information under our belts. We can start predicting and expecting what's going to happen in the next couple lectures. We're going to continue to look at development and growth in eukaryotes and prokaryotes, how they divide. And I want you to start building me a big table. So for now, just give me on a side piece of paper 2 reasons why eukaryotes have to go through mitosis, but prokaryotes don't. And then I want you to build me at the end of this lecture, I want you to build me a table of all of the stages of mitosis, but leave a lot of room because we're going to build that table bigger when we get to meiosis. And I want you to be able to have space to compare and contrast all of them. A bit of a review. I know you're probably more worried about cell respiration than mitosis right now. Totally fair. I put some notes about exam 1 here. We'll talk about this in my review. I'm reviewing my cellular respiration on Monday and Tuesday of this week of the exam. I'll do mitosis and meiosis on Wednesday and Friday of the week before the exam. So this is a hypothesized diagnose. That means we're going to give you a system and then ask you a what if it breaks in a particular location. Here is cell respiration with no oxygen. Now, the answer for many of these hypothesized diagnosed questions is it dies. I don't want that as the answer. I want to know why it dies. So here, we had talked about this in the last lecture. There's no oxygen. What can still happen? So what part is anaerobic? What part is aerobic in this system? And we'll talk about it in review. Finally, the cell cycle. I'm going to do this in 3 parts. It's actually a pretty short lecture because I know you're coming back after the exam when you're really preparing this. You do have workshop the week after the exam. You do have a quiz that day in workshop. And you also have, if you're watching this a little bit early, you also have this week, you have your career assignment due. You also have a quiz as well. So there is a quiz every single week, no matter what else is happening. You have to take that. It's the practice questions. You have to go after workshop into the practice question button in Sakai and press it, and you have 10 minutes to take that. Please come and see me if that's new information to you. I think everyone's taking it, but just in case, I want to be sure. I did find this out. I found this post that the mitochondria in October turns into the phytochondria. So that just makes me feel like it's 2020. Let's get started. Genetic material needs to be organized. We talk about it like it's genes and it's little tiny discrete pieces of information, but it's very large. If I took out your DNA and I spread it out, it would go all to the moon and back lengthwise. You are incredibly packed full of information. And I think the joke is you are neither small nor insignificant. You're just incredibly well folded. There's a massive amount of information in every single one of your cells, but it's tiny and wrapped up so tight that it doesn't look like it's much, but it's incredibly significant. The idea here is we need a way to replicate that information. Cell division. I take a cell, the basic unit of life, the thing that first evolved and became life. I take it and I have to have it reproduce and make accurate copies of itself. It needs to pass on its own information so the babies are making the same proteins as the parents. We call this the cell cycle, and it's a cycle because just like with the Calvin cycle and the Krebs citric acid cycle, we do something and then we go back to the beginning. So the cell cycle for eukaryotes, this is mitosis. The cell cycle is how we get around and reproducing each new generation. So there's 3 functions to the cell cycle. I think I've said most of them in the last couple minutes. Normally I'd have everybody yell this out, but I mean, you can absolutely yell out the answers. Yeah, absolutely. So we have reproduction, development, and growth. And this is the one that always comes up last, so I put it last, is tissue renewal. When you skin your knee or you get a little cut on your hand, your cells have to repair themselves. And what we mean by that is new, healthy, fresh cells grow. Your surviving cells that didn't get torn up divide, and they replace the ones that were killed when you fell on your hand or skinned your knee. So inside of that cell, we've seen these pictures of eukaryotes. I put a little prokaryote here. You have all the DNA, and in a eukaryote, that's contained within the nucleus. In a prokaryote, it's contained in the nucleoid. And remember, there's no membranes inside of a prokaryote. Prokaryotic cells have a single circle of DNA. Eukaryotic cells have several linear, and linear means lines of DNA, and they may have many of those. We have 46, but some of the ferns and some of the aphids, little tiny green insects, have hundreds of linear chromosomes that they need to keep organized and pass on to their offspring. We kind of talked about this before. I want to go way more in depth now. We talk about it in 2 ways. As chromatin, This is sort of the loose DNA. It's right here, this squiggly bit, and all of the proteins mixed in. These are going to be proteins that help it keep wrapped up, but there's also other proteins inside the nucleoplasm, the goo inside the nucleus. When we say chromosomes, some means body. When we say chromosomes, we're saying that the DNA and the proteins are packaged tight. You can see chromosomes with a light microscope. And in some cases, in the salivary glands of something like a fruit fly, they're so big, you can actually see them with a magnifying glass. They're packed tight and they're wound up so tightly that they become thick and a little bit more visible. Chromatin with a regular light microscope or your eyes just looks like goo. Soup. Eukaryotic chromosomes, and I'm primarily going to talk about eukaryotes today. Eukaryotic chromosomes consist of DNA and proteins. So here I've got some DNA, and this DNA is wrapped around, this blue is the DNA here. This DNA is wrapped around proteins called histone proteins. Histone is the name of the protein itself. Histones act as a way to wind up the thread. So I'm going to pause here. I absolutely have this in my house. I always wish I brought this to lecture, and here I am in my house. Give me a minute, and I'll be right back. So if this is my DNA, I got this, and I mean, I've just picked up some yarn in my house. But if I have this yarn, and the cat's playing with it and stuff, this is really unwieldy, right? Can you imagine? This is just a little bit of the length of the information of your DNA. And here, if it's out like this and it's moving around in your cell, it's going to be everywhere. So we can't have your DNA wrapped up straight up to the moon length. It's got to be packaged into your nucleus. So what you do is you have some proteins. I don't actually have a spindle. I don't weave. I just have yarn. Histone proteins act as a way to organize the DNA and keep it nightly wound and organized here. Look at all this thread that I just had, all this yarn that was all over the place. And here I've wound it around my histone forks in a nice tight way. And now it's very organized. In this example, my forks are histones and the yarn is DNA. Now there's a problem here. I've got my wound up DNA and I got a little bit loose here. When it's wound up tight like this, the proteins that you use to read the DNA and make messenger RNA out of it, to use it, can't get in here because it's so tightly wound it actually can't fit. So really tightly wound parts of DNA are difficult to read. In fact, it might be impossible to read. When you keep your DNA loose as chromatin in your cell, you keep some of it wound up, but the part that's loose, this little bit here, is the part that cell is using. This part is not. When you get ready to go through the cell cycle and you want to prepare your DNA to move around, to divide into two separate cells, you wind it all up. It's called condensation. It gets smaller and smaller. Well, it gets more condensed until I end up with a chromosome. DNA wrapped around the histone forks, coiled up really tight into little containers that we might even see, many histones wound together. I don't have that many forks, would be in nucleosomes. And then as I continue to condense it, I condense it ultimately into a chromosome, just one of these, and then it's much easier to copy and move around, or move around. It's already been copied. Chromosome number has to do more with uniqueness of the species than it does with how big you are. Now, the example I'm going to give here, humans have 46 chromosomes. That's 46 lines of genetic information, and each one of those is full of hundreds of thousands of pieces of adenine and cytosine, the building blocks, the monomers of DNA. It's full of hundreds of thousands of genes, unique codes for specific proteins. And together, those 46 chromosomes inside of you and me and everyone human watching this video, that gives us the unique humanness to our proteins. It's what makes us humans. Literally, it makes us human. Elephants have 56 chromosomes, not because they're bigger, although they are, but because they have a different set of information organized in a different way. A lot of it's the same as ours, but a little bit is different, and that's what makes the difference between an elephant and a human. Now, I drew all my own pictures for these lectures. I think you should too. I've looked online. There's a lot of beautiful pictures, but nothing quite represents you making it yourself. So I drew my own picture here. You can draw your own. This is a chromosome. Now it's already wrapped up tight. So it's wrapped up around its histones and it's wound up completely tight. And if I looked under a microscope, maybe I could see it. But you're never going to see one like this. It's just never going to happen. It's too pretty. A couple vocab words that you're going to need to know. We're going to talk about ploidy a lot. Ploidy is the number of the sets of chromosomes. In a mosquito has 2 sets of chromosomes, one set of 3 and another set of 3. That makes a mosquito 6 chromosomes. You have 46 chromosomes total, but you have 2 sets. You have one set of 23 and another set of 23. And you express them together, that's what makes you unique, and you have inherited those from your parents. Now, when I just have one set, 23 in our case, or a set of 3 for a mosquito, we call that condition haploid, or the ploidy is one set. A haploid cell, you could represent this as N, where N literally stands for the number that is in a set. So it literally stands for some number. It means a haploid cell has one set of genes, a full set of chromosomes. Sperm, egg, gametes in humans, in mosquitoes, in malaria contain haploid cells, but the rest of your body contains diploid cells. Di is 2. You have 46 chromosomes, one set of 23 and another set of 23. You got those from your parents. Your parents passed on a gamete and another gamete to you, 2 haploid cells. Your father gave you a haploid sperm. Your mother gave you a haploid egg, 23 and 23. And when they got put together, fertilization, they made you 46 chromosomes. You're made of a set of mom and a set of dad. Your body cells, your somatic cells, are all diploid, and that is true for most of all animals except for honeybees. And we'll talk about that. It's a good story. Now here, when I look at the genes you got from mom and dad, here's a chromosome that you got from mom and a homologous chromosome that you got from dad. Each chromosome, if they're matching pairs, have the same genetic information on them. So mom gave you a pink gene for earlobe shape and dad gave you a blue gene. Different types of shapes of earlobes, but both gave you some information for your earlobes. Mom and dad also gave you genes for eye color. And in this case, they both gave you the same information. Mom gave you pink eyes, dad gave you pink eyes, and so this is the only choice you have inside of your genes. Genes are just instructions for a protein. So when I talk about you making your genes or getting your genes from your parents, that means you got your genetic instructions from them. Now, every chromosome is going to have this little pinched in area. It's the centromere, the mere little spinchy bit in the middle. Centromeres are, they're not necessarily in the middle, but they're going to be very important in how we move these around. in your cell. So here, this chromosome right here, here's a chromosome. Prior to mitosis, prior to dividing the cell, your DNA is doubled. So you take a chromosome and you go through DNA replication, you copy all of the information and you make an identical copy, and you end up with 2 identical copies of all the information, and they're right next to each other. They stay kind of stuck together. That ends up looking kind of like this. They're identical copies. The number of sets of information you have does not change just because you made an identical copy. The ploidy doesn't change. The ploidy here is a single chrome of a single set is still a single set in this picture. It's the same genetic information. There's nothing new, so it's not a different set. Think about this as like a set of textbooks or a set of, any book that you have. When I bought my first set of copies of Lord of the Rings, my Lord of the Rings books had, a hand-drawn cover on them, and they were written for an American audience because that's where I bought it. That is my set, my set of Lord of the Rings. Later on, I bought another set in Britain, and that set had different spelling. Some of the information was different. Some of the words were worded differently. It's a different set, but the same general idea. Those represent 2 sets of information. Just the same exact copy of Lord of the Rings over and over again is nothing new. It's just a more mass of the same information. But sets with variation, that's something different and something new. This represents more of copying identical information. This is nothing new. So no new sets, no new ploidy. So when I take my chromosome and it doubles its information, it gets DNA replicated, I end up with 2 what we call sisters. They're sister chromatids. And this means they're little chromosomes. These sister chromatids form, and together they still make up 1 chromosome. Don't think about this too much. It'll make you lose your mind. And after a while, it'll just come and make sense to you. Just start with replicating the DNA does not change the ploidy. It just changes the mass. Sister chromatids make up a chromosome as long as they are stuck together. So a question for you about parts, and you might see something like this on the exam. Remember, we ask you to sequence and compare and contrast, but you also need to know what all those words mean. Here's a practice question for you. I want you to identify the structures here. W is a blank, and Y is our blank. And I'll see you back in the next video. Bye-bye.